Thank you for those who joined back for this talk. I guess it's a little bit less stressful with less people, so I guess that's the upside. I'm Steve Reyntjens. I'm with Thermo Fisher Scientific, responsible for product marketing for some of our Cryo-EM products, and I also speak for my Thermo Fisher colleagues. It's a great honor and a pleasure to be part of this event, so thank you everyone for having us. I don't have, obviously, I don't have 50 years of EM to look back onto, and I actually started my career in electron microscopy on the ion side. And when I was preparing for this, I realized it is almost 30 years ago that I started first using a FIB during my PhD work at Leuven University in Belgium, and that was using a Micron 50 kilovolt Gallium FIB. And I think we were probably one of the first groups, at least in Europe, who were using FIBs back then for applications other than semiconductor. So we were doing fabrication, modification of microelectromechanical systems. No cryo back then. And then a few years later, I found myself in FEI in Eindhoven. Started there in 2002, and pretty soon we were starting to experiment with cryo-dual beam. So from the single beam FIB to the FIB-SEM with Alto 2500 cryo stage at the time with people like Mike Hales then, and really very basic experiments. We were just trying to determine the milling rates of ice under 30 kilovolt gallium. And it was very interesting to see that it was about 100 times faster than silicon, which is what everybody was used to back then. And then a few years later, I have a very fond memory of an early CryoFIB workshop in Coleraine in Northern Ireland that was organized by the University of Ulster. And you see a younger version of myself there. Mike Hales was there, Bruno Humbel, Mike Marco, Alex Rigord, who was with Professor Baumeister's lab at the time. And so that was really a culmination, an exchange of experiences. It was still very early. But I think in that workshop, when we were demonstrating the dual beam with the cryo stage, we probably did one of the very first cryo slice and view experiments there. It was still rudimentary. It was not a biological sample, but yeah, a little bit of a pioneering feeling looking back on it. Fast forward something like 10 years, and FEI Thermo Fisher working towards more dedicated cryo-FIB systems. The Aquilus product, gallium-based FIB, the first dedicated cryo-FIB, and then soon followed in 2019 by the Aquilus 2, where we added the cryo lift-out capability, the integrated light microscope, and also the start of automation. I think very important to, especially to create cryo-Lamellae, the automation. Still a bit clunky maybe in the beginning, but that did come a long way, and I think now pretty much everybody's using that, and some labs are creating 50, 60 lamellae in a 24-hour session with that automation. Then in 22, a bit of a paradigm shift with the Arctis product, a fully enclosed CryoFIB, a lot of TEM technology in there, right, with a very small specimen chamber, basically a modified octagon, an autoloader for easily exchanging samples between the FIB and the TEM, and a plasma FIB. So our first cryoplasma product. And then followed in 2023 by the Hydra Bio, back to a more traditional setup with a large chamber, but a versatile tool that you can also use for volume EM, be it in cryo or room temperature. On the top of the timeline, some key publications about method development and instrumentation development in academia. it's by no means complete, of course. There's a lot more, but some really critical things that also found and potentially will find their way into commercial products to make them available to more people, which I think is useful. If you zoom out, and I think Chris had a similar graph in one of his charts, looking at the electron microscopy data bank and the number of depositions per year. The dark line is single particle and the lighter line is cryo-ET. So both, of course, exponentially growing. We all know that by now. And it looks like tomography is really on a similar growth trajectory as single particle was something like 5 or 6 years ago. So that's quite exciting. And from our vendor perspective, we also think, of course, in terms of instruments and so on. So we like to track, or we like to understand adoption of tomography in cryo-TEM labs. And so we think that by now more than half of the cryos labs also have a cryo-FIB and are doing tomography. So you see that's also been growing very steadily by 5, 6, 7% each year over the last 5, 6 years or so. Maybe an interesting topic is gallium or plasma. Of course, a couple of years ago, there was pretty much only gallium for cryo-FIBs, although plasma FIBs have been introduced for material science applications maybe something like 15 years ago, I think already. We didn't really use that much in life sciences. And then when we introduced ArcTIS in 22, you see a steep takeover almost by plasma. So plasma is becoming very wanted, and last year even more customers were preferring plasma over gallium FIBs. The difference is obviously in the ion that you use to do the work. Gallium FIBs are generally better at creating a very tight spot at low currents. I'd say that's not so super important for making TEM lamellae because you're basically using the edge of the beam mostly to shave off material. If you want to drill small holes, it's very important, but that's not what we do in life science. And then with the plasma FIBs, because the ions are extracted from a plasma, the plasma is created from a gas, you can relatively easily have different species in your focused ion beam. Xenon, argon, oxygen, nitrogen are the ones in the Thermo Fisher equipment. And xenon is probably the most relevant to cryo applications. It's an ion that's quite a bit bigger than gallium, which makes that the penetration in the material is lower, so you're expecting a bit shallower damage layer. And it also has a higher sputter yield because of the bigger ion. Plasma FIBs can also create much more current at the high end of the range, which is nice if you want to remove a lot of material. For instance, when you're using the waffle method or other high pressure frozen samples need to be machined. I'm going to zoom in a little bit more on the Lamella prep, and I think this is probably well known. So the movie shows a cell on a grid, and with the FIB you're going to remove material on top and below of your area of interest, leaving behind a thin lamella that can then be taken to the TEM for data collection, for tilt series collection. And you would be maybe thinking the thinner the better, but thinner is not always better. And I stole the title from a paper from Martin Beck's group at the Max Planck in Frankfurt. Of course, you want to be thin to avoid excessive inelastic and multiple scattering in the TEM, which will cause signal loss. But on the other hand, we know that on both sides of the lamella, there will be a damage layer, and that damage layer can obscure, let's say, the pristine part in the middle if that gets really thin in comparison. So from that aspect, you don't want to go too thin, and there's probably an optimum somewhere in between. Now, what thickness can we expect? I think it's fair to say that with the automation, 150 to 200 nanometer can be routinely achieved. And the slide shows four distributions, thickness distributions, if you will, from 4 different papers, 2 with xenon plasma FIB, 2 with gallium FIB. And you can see the median of the distribution is always in that 150 to 200 nanometer range, with some outliers, thinner and thicker, of course, but the majority will be typically in that range. It says, and I said thickness, but I think it's also important to realize that it's local thickness, so it's tomogram thickness, if you will, not necessarily lamella thickness, because the lamellas are typically wedge-shaped. So they will be quite a bit thinner on the top where the ions first hit, and then they wedge out and become wider at the bottom. And the difference can be as much as 100 nanometers. Now, I'm not sure if that's a problem. It seems to be okay. I'm also quite sure that it can change, and we can make these more parallel if we want. For instance, in hard materials, we make lamellae that are much thinner, like tens of nanometers, and they can be made quite parallel. So I think there are ways to make this more parallel if it was needed. Back to the damage for a second, and the ideal sweet spot, let's say, what's the best sample thickness? The chart here is from a recent preprint that literally came out 2 weeks ago from a big collaborative project where many people worked together and a lot of data was acquired at Thermo Fisher in Eindhoven and in Brno in the Czech Republic. It's Chlamydomonas cells on grids, more than 1,800 tomograms. And what you see in the chart is a B-factor plot of sub-tomogram averaged ribosomes versus thickness of lamella, or local thickness of lamella. And you see, of course, on the high end of the thickness, the B-factor goes up because of the inelastic scattering. But also, if you go too thin, it goes up again because of the impact of that damage layer. And so, in this case, the authors described that the best lamella thickness is between 125 and 225 nanometers. So quite achievable. There's been a lot of work and study around these damage layers in material science, and silicon is very easy. So I just want to show one data point here about what we can learn from silicon and reducing the acceleration voltage of the ions. So in silicon, it's quite easy. You can just create a FIB milled edge, like one side of a TEM lamella, then create an actual lamella perpendicular to that and stick that in the TEM. You can really literally observe your damage, which is where the crystal lattice is disturbed. And if you go from 30 to 5 to 2 kilovolts, the thickness of that damage layer is reduced by as much as a factor of 5. And that's true for gallium FIB as well as for plasma FIB. So that can be very interesting. And remarkably, I don't think many people are doing that in cryo or with life science samples. There's one paper that I know of from IBP where they've looked at reducing the voltage from 30 kilovolts to 8 kilovolts with gallium, and that gives a factor 2 reduction already in damaged layer thickness. So I think that's an interesting area. Of course, if you're going to reduce your acceleration voltage, you pay a small penalty in milling speed. And of course, you would have to add a step to your whole process. You don't want to do the entire preparation at this low energy, but you only want to do the very end, the last polishing steps. So that would add probably a couple of minutes to your preparation process. I want to switch gears now and talk a little bit about volume EM and more specifically cryo volume EM. This is basically from that same preprint that I just mentioned, and you can see the collaboration team on the slide here. What you're seeing is an entire Chlamydomonas cell sliced with the FIB, and then each slice is imaged, so it's a serial sectioning technique. All of the data, by the way, the slice and view as well as the tomograms are available on Empire, and the tomograms are also on the CZII Cryo-ET data portal. And this slice and view technique, this volume EM, being able to look at an entire cell is really nice to provide context, to help navigate what you're doing with the very high resolution stuff. And I think it can also be a way of targeting. So you can imagine slicing through a certain volume, and when you observe a specific event or a structure, you can stop the slicing and then create a TEM lamella there and go to the higher resolution tomography. This is all described very nicely in that preprint. So there's the 3D volume, and then lamella overview, and lots of high-resolution tomograms. This is how that slicing view looks in raw data. It's not really raw. This is a bit digitally enhanced, but this is how it essentially comes off the microscope. And so these are 335 slices, 20 nanometer in between. Takes about 6 hours to acquire. This was done on the Hydra Bio with the plasma FIB. And so it's a vitrified cell. There are no stains in there or anything. So it's as close to native as you get, I guess. Last piece. So it's great. I think it gets a lot of interest. But whatever you do, there will be some artifacts. It's not perfect. And charging is really a worry. On this data, it's not too terrible. It depends really on what you're looking at. It can be worse. It's usually not obstructing identification of what you want to look at, but it can be quite challenging for segmentation. With the SEM, there are tens of parameters to play with, so there is a huge space that you could explore, and I think that will be really a topic of research in the near future. But one very interesting thing that I'd like to show is some work from the Rosalind Franklin Institute in Oxford, Maud Dumou and team. And what they've looked at is, instead of the typical raster scan that you do for an SEM image, so basically going from left to right, line by line, they've come up with a much more sophisticated scheme to scan, if you like, the area of the image and distribute the dose in a much more even way, which really helps with charge dissipation. And they have really shown in their preprint from last year also some really exciting results. The left and the middle here are like traditional raster scanning, and the right is their experimental interleaved frame integration setup. And I think, yeah, the images speak for themselves. There's hardly any charging visible, and obviously you can see a lot more detail. And here's a second similar example. Again, the interleaved frame integration approach on the right-hand side, this time on a tissue, high-pressure frozen tissue sample. And with that, I hope I've tried to show a couple of things about FIB. It's by no means a complete review, of course. I think there will also be other people talking about that later this afternoon. This is the preprint. I think it's a very exciting next step in cryotomography and CryoFIB. So thank you very much for listening. I don't have a chairperson, so should I ask if there are any questions, perhaps? Oh, Wai's going to help me. Thank you. Thanks a lot.
Any questions? Maybe I can ask the first questions, which is in your slice and view types of operations, what kind of resolution you can resolve the details?
Of course, it always depends on many things. But
depends on, say, if I want to look at organelles standing next to the next organelle, I want to see anything in between.
The data that I've shown here has a 5 nanometer pixel size. That's not necessarily the resolution then that you find,
right.
But I think it's going to be in that order, 5 to 10 nanometers, depending on what type of sample you're looking at and what conditions you're using.
Okay, that sounds pretty good. Any other questions? Okay.
Thanks, very interesting. We have a Hydra Bio, I work with WA, and we definitely struggle with charging artifacts, and I'm interested in this interlead frame integration. We did find the tab on our interface with XT, but we don't see any integration with Slice and View, with the software interface.
It's really experimental at the moment. It's not built into the product at this moment.
Okay.
So it's something that's being explored, and I think it will probably end up in a product at some point.
But ideally, one would like to have that integrated with Slice and View. Yeah, thanks.
Thank you.
Maybe you need to make it public before that can be published.
Okay.
Any other questions? Okay. Do you think some of the lower voltage TEM can be useful to look at? Some of the 200 kilovolt microscope could be useful to look at for tomography?
Yes, certainly. Of course, the thickness of the sample is going to be a more important challenge, I guess, than at 300 kilovolt. But I think certainly if you're not going after the highest resolution, for these overviews of what's happening in a cell, yeah, I think it can be definitely useful. There have been some publications as well.
Okay. If you want to target what Chris is talking about, lower the cost for some poor people.
Okay. Yeah.
Okay. Is any other point? Or if not, let's thank Stephen and also the Thermo Fisher for their support. Thank you.
Thank you.